Right now, without asking your permission, something inside your chest is moving. You did not start it this morning when you woke up, and you will not be the one to stop it tonight when you fall asleep. Place 2 fingers on the side of your neck, just beneath your jaw, and wait. There it is. A pulse. A small, rhythmic shove of pressure traveling outward from a fist-sized lump of muscle that has been working since roughly 3 weeks after you were conceived, long before you had a brain capable of thought, long before you had lungs capable of breath. Let that settle for a moment. Your heart began beating before you existed as a thinking creature, and it will likely be the last organ to stop when you cease to be one. This is the paradox we are going to take apart today, piece by piece, cell by cell. Your heart is a muscle, and muscles are things you control. You decide when to lift your arm, when to clench your fist, when to walk across a room. But you have never once decided to make your heart beat, and no amount of willpower will make it stop. It is, in the truest engineering sense, an autonomous machine, a self-igniting, self-regulating electrical generator that happens to be built out of meat instead of metal. And like any honest piece of engineering, it does not run on magic. It runs on physics. Specifically, it runs on the movement of charged particles across microscopic membranes, repeated in a cascading wave roughly 70 times every single minute of your life, around 100,000 times a day, close to 3 billion times across an average lifetime. Look at your own chest for a second. There is no switch in there. There is no central command center sending out a manual order with each contraction. Instead, buried in the upper right chamber of your heart, in the right atrium, sits a tiny cluster of specialized cells called the sinoatrial node, often shortened to the SA node. This cluster is smaller than a grain of rice, and yet it is the single most important pacemaker most people will ever own, generating an electrical spark entirely on its own, with no outside command required. Cardiac cells, unlike almost every other cell type in your body, possess a strange and beautiful property called automaticity. They do not wait for a nerve signal to tell them to fire. They are inherently unstable in the most productive way possible. Their internal electrical charge slowly drifts upward on its own, like a bucket filling with water, until it reaches a threshold and spontaneously discharges, sending a jolt of electricity outward into the surrounding tissue. This is the philosophical crack in everything you thought you understood about your own body. You assume there is a you in charge, a conscious driver behind the wheel of this biological vehicle. But the engine that keeps that driver alive does not consult the driver at all. The sinoatrial node fires whether you are thinking about calculus, falling in love, or fast asleep with no thoughts at all. It is, quite literally, a piece of you that operates entirely without you. Once that spark fires in the sinoatrial node, it does not simply explode outward in a chaotic burst. It propagates in an extraordinarily disciplined wave, racing across the muscle fibers of both upper chambers, the right and left atria, at a speed of roughly 1 meter per second. This wave causes the atria to contract almost simultaneously, squeezing blood downward into the 2 lower chambers, the right and left ventricles, through one-way valves that snap shut the instant the pressure reverses, preventing any backward flow. This is the first half of the sound you have heard 1,000,000 times in movies, and in your own ears when you press a stethoscope or a fist against your chest. The soft, low lub. But here is where the engineering gets genuinely elegant. If the electrical wave simply continued straight through from the atria into the ventricles at full speed, the upper and lower chambers would contract at nearly the same instant, and the ventricles would try to pump blood before the atria had even finished filling them. The system would short-circuit itself through poor timing. To prevent this, evolution built in a deliberate, almost frustrating delay. The electrical signal, after racing across the atria, reaches a second clump of specialized tissue called the atrioventricular node, or AV node, sitting at the junction between the upper and lower chambers. Here, the wave is forced to slow down dramatically. Conducting at a fraction of its previous speed, creating a pause of roughly 100 to 120 milliseconds. That pause is not a flaw. That pause is the entire reason your heart works as a pump and not as a useless simultaneous twitch. After that pause, the signal is released into a remarkable structure called the bundle of His, which splits almost immediately into left and right bundle branches, fanning out into a dense, tree-like network called Purkinje fibers. These fibers are the fastest conducting tissue in your entire heart, built almost like an electrical wiring harness, specifically engineered to deliver the impulse to the very bottom tip of the ventricles first. This is crucial. The contraction needs to begin at the apex, the bottom point of the heart, and roll upward, so the blood is squeezed efficiently upward and out through the great arteries, rather than sloshing uselessly side to side. When this wave hits the thick, powerful muscle of the ventricles, they contract together with tremendous force, slamming the valves between the atria and ventricles shut, producing the second and sharper sound, the dub. Lub, dub. Two sounds, two distinct mechanical events, separated by a carefully engineered electrical delay measured in milliseconds, repeated without your permission roughly once every second for your entire life. Consider the sheer mechanical reliability this demands. A man-made pump engineered to perform a single repetitive motion 100,000 times a day would be considered a triumph of industrial design if it lasted a single year without failure. Your heart is expected, by the simple fact of your continued existence, to perform this exact sequence without meaningful interruption for 70, 80, 90 years, contracting roughly 3 billion times across a single human lifetime, built from tissue that, unlike skeletal muscle, almost never regenerates new cells once you reach adulthood. The cardiac muscle cells beating in your chest right now are, for the overwhelming majority of them, the very same cells that began beating while you were still an embryo the size of a grain of rice. They have never rested. They have never been replaced. They have simply continued, generation after generation of electrical impulse, asking nothing of you in return except a steady supply of oxygen and glucose carried in by the very blood they themselves are pumping. There is something almost unbearably elegant in that closed loop. The engine fuels its own fuel supply, indefinitely, without complaint. And we have not even begun to discuss what is actually happening inside each individual cell to make this electrical spark possible in the first place. We have not discussed the sodium, the potassium, and the calcium ions flooding in and out of microscopic channels with the precision of a Swiss watch. We have not discussed the men who spent their lives, and in some cases sacrificed their sanity, trying to capture this invisible electrical storm on paper for the very first time. That is where we are going next, into the substrate beneath the sound, into the cellular machinery and the historical obsession required to ever prove that your heart runs on electricity at all. To understand what is actually happening inside a single heart muscle cell, you need to forget the idea of a switch turning on and off. Instead, picture a dam holding back a reservoir. Every cardiac cell maintains a voltage difference across its outer membrane, roughly -90 millivolts on the inside relative to the outside, held in place by pumps that constantly shove sodium ions out and potassium ions in, fighting against the natural tendency of these charged particles to spread out evenly. This negative resting voltage is potential energy, water held behind a dam wall, waiting for a trigger. When the electrical wave from a neighboring cell arrives, it nudges this voltage upward just enough to open a gate, and that gate is a sodium channel. Sodium ions, which are vastly more concentrated outside the cell than inside, come rushing in through this opened gate in a flood that takes well under 1 millisecond, slamming the voltage from -90 millivolts up to roughly +20 millivolts almost instantly. This is the dam breaking. This sudden reversal is called depolarization, and it is the trigger for everything that follows. As that wave of depolarization spreads from cell to cell across the heart, it does not have to jump through empty space the way a spark jumps across a gap.
Heart cells are physically connected to their neighbors through specialized junctions called gap junctions, tiny tunnels that allow ions to flow directly from the inside of one cell into the inside of the next.
This connectivity is what allows the entire heart to behave less like a collection of separate cells and more like a single unified electrical sheet, a property physiologists call a functional syncytium. This unified sheet is not merely an elegant design choice, it is the entire reason a heartbeat remains orderly instead of collapsing into chaos. If those gap junctions were ever disrupted, or if a rogue cluster of cells somewhere in the ventricles began firing its own competing rhythm out of sync with the sinoatrial node, the coordinated wave would fracture into dozens of disorganized, simultaneous wavelets, each one triggering its neighbors at random, producing a chaotic quivering rather than a single powerful squeeze. Physiologists call this fibrillation, and it is the clearest possible demonstration of what this entire electrical architecture is actually protecting you from. A heart in fibrillation still contains every one of its individual cells, every one of its ion channels, every drop of the calcium needed for contraction. What it has lost is coordination, the disciplined, one-directional timing of the wave. And coordination, it turns out, is the entire difference between a functioning pump and a useless, twitching bag of muscle. But depolarization alone does not cause a muscle to contract. That requires a second messenger, and that messenger is calcium. As the depolarization wave passes through, calcium channels open more slowly than the sodium channels did, allowing a trickle of calcium ions into the cell. This trickle is the trigger for an even larger release. It signals an internal storage structure called the sarcoplasmic reticulum to dump a massive flood of additional calcium into the cell's interior. This flood of calcium binds to the contractile proteins, actin and myosin, allowing them to grab onto each other and ratchet past one another, physically shortening the muscle fiber. This is the actual mechanical contraction, the squeeze, happening at the molecular level, triggered entirely by calcium concentration. Once the contraction is complete, potassium channels open, allowing positive charge to leave the cell and restore that negative resting voltage, a process called repolarization, resetting the dam so it can be triggered again on the very next beat. This entire sequence, from the initial sodium flood to the final potassium reset, is called the cardiac action potential, and it takes somewhere between 200 and 400 milliseconds to complete in a single ventricular cell, repeating with almost flawless precision, tens of thousands of times a day for decades on end. The fact that this process even has a name, the fact that we can describe it in this level of mechanical detail, is the result of an intense, decades-long human obsession to capture something that, for most of history, was completely invisible. That obsession found its breakthrough through a Dutch physiologist named Willem Einthoven. Working at the University of Leiden in the final years of the 1800s, at the time, scientists already suspected that the heart generated tiny electrical currents, but the instruments available could only barely detect them, and certainly could not record them with any precision. Einthoven spent years refining a device called the string galvanometer, a delicate instrument using a thin, silvered quartz wire suspended in a powerful magnetic field. When even the faintest electrical current from a beating heart passed through that wire, it would deflect, and that deflection could be photographed, frame by frame, producing the first reliable tracing of a heartbeat's electrical signature ever captured by human instruments. Einthoven named the distinct waves he observed P, Q, R, S, and T, a labeling system still used by every cardiologist on the planet today. And in 1924, he was awarded the Nobel Prize for what we now simply call the electrocardiogram, or ECG. Before Einthoven, the heart's electrical activity was a rumor. After Einthoven, it was a measurable, repeatable, visual fact. Around the same period, a Swiss-born physiologist named Wilhelm His Jr. was performing meticulous dissections on embryonic hearts,
searching for a physical pathway that could explain how electrical signals seem to travel from the upper chambers to the lower chambers in such an orderly fashion. In 1893, he identified a slender bundle of specialized muscle fibers running directly through the fibrous tissue separating the atria from the ventricles, a structure that now carries his name, the bundle of His. This was the missing highway, the physical bridge connecting the AV node's deliberate pause to the rapid fan-out of the Purkinje fibers below. Decades of painstaking, often poorly funded anatomical work by His and later researchers like Sunao Tawara, who mapped the AV node itself in 1906,
slowly assembled the complete electrical map of the human heart, piece by piece, one obsessive dissection at a time. What is remarkable is how mechanical, how almost dispassionately physical all of this turns out to be. There is no ghost in this machine. There is no separate vital force animating the tissue. There is only sodium rushing in, calcium triggering a molecular ratchet, potassium resetting the charge, and a precisely delayed relay system built from specialized fibers, discovered by men hunched over microscopes and crude galvanometers, slowly dragging the invisible electrical storm inside your chest out into the light of measurable science. And yet, knowing every one of these mechanical details still leaves one enormous question unanswered. If the sinoatrial node fires on its own, with no outside command needed, who decides whether it fires fast or slow? Who decides whether your heart races during a moment of fear or settles into calm stillness during sleep?
That answer lies outside the heart entirely, in a tug-of-war happening inside your nervous system, and that is exactly where we are headed next. The sinoatrial node generates its own spark, but it does not generate that spark at a fixed, unchangeable rate. Left completely alone, disconnected from any outside influence, an isolated human sinoatrial node will fire on its own at somewhere around 100 beats per minute. That number is worth sitting with for a second. Your natural, intrinsic, untouched heart rate is not the 60 to 70 beats per minute you probably associate with a resting adult. It is closer to 100. The reason your resting heart rate sits so much lower than that is because of a constant, deliberate braking force applied by your nervous system, specifically by a long, wandering nerve called the vagus nerve, which releases a chemical messenger called acetylcholine directly onto the sinoatrial node,
slowing its natural firing rate down through a kind of perpetual restraint. This braking mechanism was discovered almost by accident, in a moment of genuine scientific serendipity, by an Austrian-German pharmacologist named Otto Levy in 1921. According to his own later account, Levy woke from a dream with the design for an experiment fully formed in his mind. Stimulate the vagus nerve of one frog's heart, collect the fluid surrounding it, and transfer that fluid onto a second, untouched frog's heart. When he performed the experiment the following morning, the second heart slowed down dramatically, even though its own vagus nerve had never been touched. This proved that nerves communicate with organs using chemical messengers, not pure electricity, a discovery that earned Levy the Nobel Prize in 1936 and named the mystery substance Vagusstoff, which simply translates to vagus substance, later identified precisely as acetylcholine. Opposing this braking force is an entirely separate system, the sympathetic nervous system, which releases adrenaline and noradrenaline directly onto the heart, accelerating the sinoatrial node's firing rate and increasing the force of each contraction. Your actual heart rate at any given moment is the constant living result of these two opposing forces pulling against each other, the vagal brake and the sympathetic accelerator, locked in permanent negotiation tens of thousands of times a day. When you are calm, the vagal brake dominates, holding your rate down near 60 beats per minute. When you are frightened, startled, or sprinting up a flight of stairs, sympathetic drive surges,
the vagal brake releases, and your rate can climb past 150 beats per minute within seconds. No conscious decision required at any point in that process.
Here is the uncomfortable modern problem buried inside all of this elegant biology. This entire system, the vagal brake, the sympathetic surge, the flood of adrenaline, was built across millions of years of evolution to respond to acute, short-term physical threats. A predator, a physical fight, a genuine sprint for your life. The system expects the threat to resolve within minutes, followed by a return to calm. But the modern world delivers something this ancient circuitry was never designed to handle: chronic, low-grade, unresolved psychological stress, repeated daily for years on end. A notification on a screen, an unanswered email, a looming deadline that never quite goes away. Your sympathetic nervous system cannot tell the difference between a genuine predator and an anxious thought spiral at 2 in the morning. And so it activates the exact same accelerator, the exact same flood of adrenaline dozens of times a day, with no physical sprint ever actually occurring to burn it off.
This is the evolutionary mismatch sitting quietly inside your chest, an ancient pump exquisitely engineered for acute physical danger, now running constantly in a low simmer of chronic psychological pressure it was never built to absorb. Fortunately, this same system gives us a remarkably precise window into how well that ancient pump is coping, and it comes from something called heart rate variability. A healthy heart does not beat with perfectly identical spacing between each beat, like a metronome. Instead, the spacing subtly varies from one beat to the next, lengthening slightly as you exhale when vagal tone naturally increases, and shortening slightly as you inhale. Higher heart rate variability, meaning more of this natural fluctuation, is consistently associated with a nervous system that can flexibly shift between the brake and the accelerator with ease.
Lower heart rate variability, a heart beating with rigid, metronome-like sameness, is associated with chronic stress, poor recovery, and a nervous system stuck leaning too heavily on the accelerator. So what is the actual protocol, the practical engineering fix for a system caught in this evolutionary mismatch? The answer lies in deliberately strengthening the vagal brake. It helps to understand precisely why slow breathing works, rather than simply accepting it as folk wisdom. When you inhale, the act of expanding your lungs mechanically stretches receptors near your heart and lungs that briefly suppress vagal output, allowing your heart rate to tick upward slightly. When you exhale, that suppression releases, vagal output surges back, and your heart rate drops slightly in response. This natural rise and fall, called respiratory sinus arrhythmia, is healthy, normal, and measurable in nearly everyone. By deliberately slowing your breathing and extending the exhale,
Typically to a count of 6 breaths per minute or fewer, you are not inventing a new biological pathway. You are simply amplifying a pathway that already exists, exaggerating the natural exhale-triggered vagal surge until it meaningfully slows the sinoatrial node within a matter of seconds. This is why a single minute of slow, deliberate breathing can measurably shift your heart rate downward in real time, a direct mechanical lever you can pull on a system you otherwise cannot consciously touch. Slow, controlled breathing, particularly extending the exhale longer than the inhale, directly stimulates the vagus nerve and increases acetylcholine release onto the sinoatrial node within seconds. Regular aerobic exercise, performed consistently over weeks and months, physically increases resting vagal tone, which is precisely why trained athletes often display resting heart rates in the 40s, their sinoatrial node held under a much stronger, more efficient brake than an untrained person's. Consistent, adequate sleep allows the sympathetic accelerator to fully disengage each night, giving the vagal brake uninterrupted dominance for hours at a time, which is something a screen-lit bedroom at midnight consistently denies you. None of this is mystical wellness advice.
It is direct mechanical intervention on an electrochemical oscillator, using breath, movement, and darkness as the tools.
And so we return to where we began, to that small, involuntary lump of muscle in your chest that started firing before you had a single conscious thought, and that will likely keep firing, unsupervised, for every remaining second you have left on this earth. It does not ask your permission, it does not consult your fears, your ambitions, or your regrets. It simply continues its electrochemical cascade. Sodium in, calcium triggered, potassium reset. Lub-dub, lub-dub. Roughly 100,000 times between now and tomorrow morning, completely indifferent to whether you spend those beats anxious or at peace. The only genuine choice you have in this entire system is not whether the beats happen, but how you choose to influence the brake and the accelerator pulling on them every single day for as long as you are given. If you made it this far, you now understand more about the electrical engineering inside your own chest than most people will ever learn in their entire lives. Leave a comment below and tell us, knowing your heart was beating at 100 beats per minute before your nervous system ever learned to slow it down, does that change how you think about stress or about calm? Subscribe for the next deep dive into the hidden machinery running your body without your permission, and we will see you in the next one.